那是什么呀，宝宝？你的测体温啊？没发烧。Okay.
Can you sit, please?
Yes,
Hello?
Dana.
Yes,
Yes,
it's
we
okay.
can
You
see
take
you.
your
Yes.
presentation now.
Okay. Good morning, everyone. Morning, Professor. Today I'm going to present my lab report. So, my name is Henrio, and my major is microbiology. So, I'm going to present what I have done and what I'm going to do and what I will do. So, in this presentation, I'm going to present three contents. So, in the first one, I'm going to present the academic class, that what is the class I have been doing for 3 months, the publication, the literature review, and the lab work on the research work. So now I'm going to present my academic class. So first of all, because my PhD start from September, so I choose and I attend 7 classes, such as integrity writing, immunology, and Chinese language, that is mandatory for the school. And then I work on my review article. So in this literature review, I have sent my article, as we can see in here. I submitted on December 23. So and I choose this journal, academic Canadian Journal of Infectious Disease and Medical Microbiology. So this journal is bi-monthly, and we hope it will get accepted at this time. This is the review article is called Zika virus and its association with neurological disorders. The research work, according to my last data, because I already discussed to professor, like I should continue my experiment or what should I do. So now I decided, I suggest if it is good for professor, if I continue my research with animal model, or what professor suggests me. So according to my data, the Zika virus protein, to my last data, Zika virus protein. So at this time I used Zika virus protein. So Zika virus protein induced necroptosis in PC12 cells, and the cell death rate showed increase after introducing Zika virus protein. And I used the Zika virus protein NS5 and PRM. So as we can see, there is a significant between two protein, NS5 and PRM. The second result I got from Western blot is the induced necroptosis in PC12 cells. And I checked the RIP1, MLKL, RP3, and the MLKL. The third result I got is the necrostatin, that is inhibitor for Zika virus protein, that is decrease the cells damage toward this protein induced necroptosis in PC12 cells. So, and the third one is I use the CCK8 detection for the alive cells. The necroptosis increase the cell survival after introducing a Zika virus. As I told, because we start from September, and I still not check whether how is my work going on. So I suggest something, if it is okay for professor, because from September till now is still 3 months. So I don't know what should I do. So I suggest the infectivity of Zika virus on primary cells using cell line or animal mice model. So I'm going to check the susceptibility of different mice primary cells to Zika virus infection. So in this time, I'm gonna use the real virus. And this one also, there is a question because as professor told me, the lab, is it susceptible for using the virus or not? I'm not sure about it. So I'm gonna use, if it is okay for professor, I'm gonna use the primary skin cells, TSDF, and primary thoracic aorta cells, the primary kidney cells, and primary lung cells. And I used to do for the last experiments, I will use the human origin cell lines, HEK 293 for human fetal kidney, the human embryonic log fibroblast, and the human hepatocellular carcinoma, and introduce them in Zika virus at the various time of post-infection and determine in RT-PCR. Then I want to know also the expression of Zika virus protein in primary TSV and TSDF cells to further confirm the susceptibility of primary TSVE and TSDF to Zika virus. The expression of Zika virus protein was detected. The presence of viral envelope or NS3 or NS5 protein in the cell will evaluate by indirect immunofluorescence assay, human stains with human convalescent serum from Zika virus patient. I want to evaluate also the expression of cytokine within the primary cells in response of Zika virus infection in order to determine whether the Zika virus induced an innate antiviral immune response in the permissive primary cells. I analyzed the K antiviral immunity related cytokine genes, genome expression change in Zika virus infected cells. The material and method. The procedure, I want to use mice model and the Zika virus and cells. The detection of Zika virus RNA in supernatant and intracellular of the infected cells. The viral RNA was extracted from cell culture supernatant using the RNA mini kit and RT-PCR kit. Also, the cell viability assay and Western blot by using CCK8 and the immunofluorescence assay in post-infection. Cell will fix with cold acetone methanol and saturate with bovine serum albumin and Zika virus human serum in blocking solution overnight. And the expression of cytokine. The cell lizard will measure at the different time of a post-infection by qRT-PCR. So, Professor, you are welcome to suggest me something whether I can continue with my research before or what should I do for it. So that's why my PPT, because I don't have any data, so my PPT is still not enough. My data to support my research is not enough because we just start from September. I'm working on my review, as you told me, to publish it as soon as possible. So I spend a lot of time to correct because they reject this 3 or 4 times. So I spend a lot of time to correct my review articles. So I send it to the journal. So we are waiting for it and we hope it will get accepted. So that is my presentation. Thank you so much.